And welcome back to the last lecture for chapter 26. So in this lecture, we're going to talk about gene therapy. So gene therapy is the insertion of genetic material into human cells for specific genetic treatments or for treatment of other illnesses. And so there are different ways that we can insert those genes into the individual. So we can use viruses, which I talked about before, where we take a virus that can infect human cells this time, genetically modify that virus so that it is no longer able to cause viral attacks, and make it be able to insert specific genes that we care about. We can use liposomes. Liposomes are microscopic lipids that we can inject specific genes into, and then we can use them to get into our cells. And sometimes we just take the gene and directly inject it into the cells. So where does this occur? It can occur in many areas of the body. So this figure shows gene transfer by injection. And so these are different diseases that can be treated using injection directly into the brain. Here we have skin cancer. Again, we're going to be transferring this directly into the skin or to the blood cells. Cystic fibrosis is we use an aerosol spray. We can inject for the liver, we don't inject, we actually implant a modified liver. So we take a liver out, a portion of the liver, not the whole liver, and then we inject the genes that we want that liver to produce into the liver, and then we put the liver back into the body. And so while you're looking at this figure here, focus on if it has 1 star or asterisk or 2. If it has 1, it is known as an in vivo. Versus 2, so that's 1 star. If it has 2, then it is ex vivo. And you may wonder, what is the difference between in vivo and ex vivo? So the term in vivo means that we are injecting or we are placing that directly into the body. So we are putting something into the body. Or cells. Whereas in ex vivo, we are taking something out of the body first. We might use stem cells or the organ itself. And then we put the gene that we care about into that organ or stem cell, and then we replace those cells. So in ex vivo, we use ex vivo methods, I should say, for treating SCID. SCID is severe combined immunodeficiency. So this occurs, it's a rare disease, but it occurs in children who are born without the ability to produce mature B and T cells. And because they can't produce those B and T cells, that's your immune cells, they're more prone to infection and they can die very easily. So with these, we use ex vivo. So we remove bone marrow from the patient. We infect that bone marrow with a virus that codes for normal enzyme production. So now we want them to produce adenosine deaminase, normal. And then we return the stem cells back to the patient in hopes that they produce normal adenosine deaminase. Familial hypercholesterolemia. So in this disease, which I've talked about in chapter 23, when we talked about heterozygosity and genetic disorders, this is a disease that affects the ability to take in cholesterol. So people that have this disease can't pull in the cholesterol. So the reason for this is the liver, which takes in our cholesterol, they lack the receptor proteins for removing the cholesterol. So what we do is we remove the liver itself, just a portion again, and then we infect it with a virus that contains the gene for the normal receptor. The liver is then returned to the patient, and we hope that it starts producing those normal gene receptors. And here is ex vivo therapy. You can see it. So here we're taking bone marrow. We're taking the bone marrow that has the defective gene. We then transmit via a virus the gene that we care about, and then what we're hoping happens is the gene that the virus is carrying is transferred to the normal genome of the cell, that stem cell. We return that stem cell to the bone marrow and hope that it starts producing normally. Then we have in vivo. So in vivo, again, we are putting something into the body directly. And so this is how we treat things like cystic fibrosis. So with that, we use a special spray that we spray down the nasal passageway. This spray contains liposomes or adenovirus vector that also carries the gene to produce normal cell function. So it produced the normal chloride transporter protein. Okay, so the gene that codes for that chloride transporter is going to be found within the liposome or the adenovirus. And then we use in vivo therapy often to help with treatment of cancer. In this case, we're going to be introducing materials into a tumor that maybe will cause that tumor to stop growing effectively, or we might make the healthy cells more tolerant to chemotherapy, and the tumor cells more sensitive to chemotherapy. So we're damaging those cells that we don't like, and we're not hurting the cells that we do like or that we don't want to damage. So that's the last slide in this video. So I'm going to post this and I'll pull these through Edpuzzle, and I will talk to you later. Bye.